Hi everyone, today I will show you how to do RNA-seq using Nanopore long reads. In the process, I will highlight the long read specific aspects of the pipeline that differ or are incompatible with standard short read analysis. Long reads are here to stay and have been a key tool for the latest paper from the lab. Here I will show you every step from getting the raw reads files to generating a table of differentially expressed transcripts. Let's dive into it. Let me walk you through the step of the pipeline that we will be covering first. We will start with 3 FASTQ samples from control samples, control 1, control 2, and control 3, and also with 3 experimental FASTQ files for experiment 1, experiment 2, and experiment 3. So this is our raw data that we are going to map to the transcriptome, and this is a FASTA file, FA. So the first thing that we do is the mapping, and what we get is this bunch of map files from control 1 to the last experimental 3, and these map files have this BAM end. Then these map files, we are going to use them to count how many of the reads map to the different transcripts. So we will count here, and we will get these TSV files from control 1 again, TSV with the number of reads for each of the transcript, and the same for the experimental till experimental 3 TSV. And finally we will do the differential expression analysis, and we will get this table for each transcript. We have the fold change, and we have the Q value or adjusted Q value. So we have transcript 1, transcript 2 till transcript N, and some transcript don't change or change on the other direction, and we have different Q values, some significant for every transcript. So, and the differential expression will be, of course, between these control samples and these experimental samples over here. With that out of the way, let's download the data from this manuscript that we recently published. We go to data availability, we click on the geo accession number. Here then we look at all the samples, and in this case we will select these 6 samples, 3 controls and 3 experimental. For each sample we are going to click on the link, then we are going to come down to the bottom and click on the SRA link. Within the SRA we are going to click on the run, then we go to FASTQ download, we select the run and download it. Then we repeat that for all the samples. We'll end up with 6 FASTQ files, 1 file for each. In this folder we have the 6 FASTQ files, and we have all the intermediate steps as well, because I already ran the pipeline, but I will rerun now all the steps for one of the FASTQ files as a demonstration. The first thing that we need to do is to map the FASTQ files to our reference transcriptome. And here is the first difference with the standard RNA pipeline for short reads. While most aligning programs are able to use short reads, only a few can also align long reads. In this example, we will use Minimap2, a commonly used alignment tool for long reads. Here we will be mapping this FASTQ file, finishing in 4. So we call Minimap2. We use the map ONT, which is the option specific for Oxford Nanopore long reads. We pass the reference transcriptome where we are going to align the reads, the name of the FASTQ file with the reads, and finally we ask to put the mapping results on a file with the same name as the run. When we launch the run, we see that the SAM file is created. Once we have that, we can move to the quantification of the reads. However, to fit the alignment to the quantification program, we have to convert the files from SAM to BAM. Hey, if you want a refresher of the basic bioinformatics files, please check the video now on the card. To convert the SAM files into BAM, we use samtools. Within samtools, we use the view function. We pass the SAM files, and we request the BAM files with the same name. So when we launch the script, we see that the BAM file is created. Hey, if you would like to learn how to automate this whole process, don't forget to check my playlist on how to use the workflow manager SnakeMake Okay, now let's move to the quantification. Here is where we have the next difference with the short read protocol. The standard RNA-seq protocols are gene-centric rather than transcript-centric. Because of this, reads are normalized according to transcript isoform length. Also, direct RNA-seq is biased toward the 3' end of the transcript. Because of this, in the quantification, we only account for multiple mappings, but not the length of the transcript isoform. In this case, we will use NanoCount, which has been specifically designed for Nanopore long reads. So we call NanoCount, we pass the BAM file, and we request the results to be copied into the TSV file with the same name. Once we have the quantification files for all the reads, we perform the DSeq analysis inside RStudio. Here, unlike standard short RNA-seq analysis, we will leverage the power of long reads to differentiate between transcripts isoforms. So instead of performing DSeq at the gene level, we will do it at the transcriptome level. First, we upload the libraries that we will use, including DSeq2, of course. Then we are going to upload the metadata from the sample.txt file, which has just the name of the runs and the conditions, either A or B. Then we will merge the TSV files generated with NanoCount. And here I'm uploading one to show you how it looks like. It has a column with transcript names and 3 other columns, but the one that we are interested in is the EST count column. In the next chunk, we will combine all the TSVs for the different runs. We now have a table with 3 columns: transcript name, EST count, and the name of the run. Next, we convert this table into a matrix where the columns are the different runs and the rows are the different transcripts. Finally, we feed this matrix to DSeq2 and we get our table with differentially expressed transcript. We then have the different transcript, the log2 fold change, and the adjusted p-values. Okay, that's it for today. Please subscribe to the channel if you like the content. And now that you have your differentially expressed transcript table, you can proceed, for example, to do an ontology analysis. For that, click the video now on screen, and I will see you there.